I welcome you all in this lecture series of Transnational Research. And the topic for this session is Prakriti Genomics. And today we have a very experienced speaker with us, Dr. Mukherjee. Dr. Mukherjee, she is working as professor in Department of Bioscience and Engineering, and she is faculty School of Artificial Intelligence and Data Science, IIT Jodhpur, one of the very
So incidentally is one of the key aspects when you try to understand Prakriti, which is the subject matter of today. So when I was young, I grew up in a very small town, but only thing which was always there is frequent displacement, which would cause a lot of stress triggers because I changed maybe 7 schools. And then I had this condition of severe sweating and did not know what to do about it. So these are all typically if you grow in a household where you have only reached to modern doctors, this is treated as some home remedy can do, some grandmother's recipe can do. And then acne, which is also unusual usual in people. And then you get into antibiotics and sometimes home remedy, sometimes Safi. And then I had bucktooth. So I had braces in my teeth. So these are like unrelated things, right? One of the other things is that I was very lean and thin, and I was very outgoing, very friendly to people, and would just not walk straight on the road or whatever it is. These are all the facts. And then in my 30s to 50s, I shifted to an urban living job, Delhi, and there I had a bleed in my brain, which required a brain surgery, and then obesity set in. So I would have been 50 till my masters, and then suddenly I became 70, and then there was an onset of diabetes and every different perspective. Now, since you are an Ayurveda audience, it's not an engaging way to ask a question, but still, I would like somebody to answer that do you think there is something pervasive in this kind of a health trajectory, or is there something which strikes you could be a driving cause? Can anybody answer? Anybody? I uh, isn't the sound enabled for the audience?
Yes, madam. Yes, madam. The sound is very clear,
Okay.
and the participants can answer in the chat box.
Okay, I cannot see the chat box myself. So are they answering? Can somebody, this is a new, ah, okay, chat box is there. So nobody is answering? So can anybody, can predict what could be the likely Prakriti or a likely Vikriti which is pervasive all throughout? No? Okay, maybe this is not the better format to ask a question. But the thing is that if an Ayurveda, yeah, so somebody has answered, very good, pith predominant Prakriti. So now you see severe sweating, acne, braces, AVM, everything is a manifestation of one underlying individuality, seems to be a pith, and maybe the next could be Vat. And so this is my maybe inherent Prakriti, which manifests as different things in different time periods, and it is actually treated in a different manner. But when uh, when I presented with this severe sweating in my 30s, I got in touch with Bhavna, with whom I worked for 20 years. 15 days of a medication of 20 years, 15 days of a medication of Ayurveda, and it cured my sweating for life. I don't have that severe problem anymore. So what struck me very clear? So what what struck me very clearly was that here is some kind of a phenotype which is very well understood in an Ayurveda domain, but not so very it's hyperhidrosis is treated in very it's hyperhidrosis is treated in particular conditions, but not seen by many doctors if you are suffering from different kinds of organ diseases. Now, such a kind of a story would be also true for many diseases, and this is like a global health and disease scenario where which was conducted across 195 countries in 354 diseases. And what came out all throughout was that though we have been able to manage infectious diseases with antibiotics and all, and we have been able to live longer, so the longevity has increased, but the last 10 years of life people live with ill health. So the health longevity has reduced by 10 years. And that's where this major fraction of non-communicable diseases are there, which causes huge number of death, and it's a very major economic burden. So the need of the hour is that we need personalized and preventive approaches for reducing such kind of a health burden. The other thing which is also getting evident in the modern scenario is that sometimes you have well-known mutations. For example, sickle cell anemia. It's caused by a defect in the beta globin gene, because of which the RBC becomes sickle in shape, which is also protective for malaria, but it causes very many disease condition. For example, people will have stroke, while people will have pain, hemolytic anemia, aplastic anemia, any one of these. So now the problem in sickle cell is not to detect the disease, because one can do a genetic testing and easily identify. But the problem is that if one is living with sickle cell disease, one cannot predict which is the morbidity where one would kind of finally be. Some can have pain, some can have bleeding, some can have stroke, and it is very uncertain who will get what. Moreover, there are environmental modifiers, for example, hypoxia, infection, or aridity, which exacerbates this. If you ask an Ayurveda doctor, then you might get to know that pain could be a crisis which could precipitate in if you have Vata perturbation, bleeding could precipitate if you have Pitta perturbation, stroke might be outcome in Kapha perturbation, aridity can cause Vata increase, infection can increase Pitta, or hypoxia can maybe increase cough. So trials are population based, and when you take it to the individual, it doesn't work anymore. So now people are moving to new paradigms. They are saying one person trial, that the same person becomes a control and a case, and you aggregate the information around by one person trial and then try to see how you can make the medicine more effective, in a sense that you can reduce NNT, so more people are effectively treated. And then there's another term called NNH, that is number needed to harm. So if that is increased, then you harm lesser number of people. The other thing which is also becoming very evident is that uh, typically modern medicine is very organ centric. So you go to a person who is a specialist in a particular organ, and even if you have diseases with the organ, it you typically get labeled into a syndrome, and that is like either a skin based or a neuro problem, or whatever the organ may be. And now what is being realized is that it is not a thing that you have one gene and causes one disease. there is a 1:1 combination possible, so that if a person, let us say with type 2 diabetes, could be let us say with type 2 diabetes could be having multiple genes contributing to this disease, and the same gene could also contribute to other diseases. So as a result of which, there could be all the diseases are linked, and that's why modern medicine is now moving from an organ system to a system specific perspective. And then there are other things which is also getting understood, is that uh, though we inherit the gene, the environment around us or the temporal variation around us contribute to a wide variability in the outcome or in the trajectory which we take. So now what people are doing is they are making data clouds around one person. So it's like a GIS, and you're looking at the patterns of people and trying to understand how does an individual respond to various kinds of trigger, and based on these responses could you bin people into groups, based on which you can identify who are the people who are similar in some kind of way, so that you have personalized therapy based out of all these observations. So this has also become a very important topic in today's day, when people are moving from one place to the other, or transiting to different kinds of diet, etc. And the whole thing now is moving from, actually, if you can have an individualized medicine right from pre-womb stage to the tomb. So, for example, before a child is born, right after a child is born, or if you can get the undiagnosed disease to prevent it, to manage infectious disease, to control cancer, or to look at differences in outcome of a particular medicine, or how you can increase the health span, or even know the cause of death. So this is the whole spectrum where individualized medicine is promising to kind of contribute from the genomics perspective. And the world is now talking about all these in the context of predictive, preventive, personalized, and participatory medicine, which is called as the P4 medicine. So how you can predict, how you can prevent, how you can personalize, and how an individual can also take a decision in managing their health. So why this whole area has got an impetus is because of this universality of our genetic code, that is the DNA. All of you might have heard about it. But what is interesting about the DNA is that it's just like you have English alphabet, the entire life form on Earth share the same common language of four chemical letters, which are adenine, guanine, cytosine, and thymine. So combinations of these in different ways is what gives rise to DNA. And all life forms on Earth, be it a bacteria, be it mouse, be it plant, be it primate, everybody has their genetic uh, code written in the same chemical language. And this whole thing, or what is called as the DNA, by all these chemical letters, is present in our nucleus. Some part is present also in the mitochondria. And if you open this up and read the whole thing, then this would be written as AGCT, millions, 3 into 10 power 9 times these chemical bases are threaded. And this is what is called as the book of life. So you can understand it better by the English letters, just like you have the alphabets, you have DNA. And if this gets threaded into words, these are called as codon in the DNA language. And if the words get threaded, what is called sentences in the language, it would be genes. And if the genes get threaded, then it forms chromosome, which is like a chapter of a book. and the entire book is what is called as the genome. So we inherit one such book from one parent, and so we have two books, one from the mother and one from the father. So now this DNA, like this, it's written in AGCT. So this DNA resides in the nucleus, but it can make a copy of itself by a method called, or a process called transcription. So it makes an mRNA. So in the mRNA, you have complementary to whatever is written in the DNA, but instead of adenine, you have another nucleotide, which is called uracil. But whatever is there in the DNA gets transcribed into an RNA, and then three of these are read as one codon, so that in threes, you have a code which is again read by another machinery called the translational machinery. So these three codons will convert into one kind of an amino acid. So if you have UGG, that means a tryptophan will be there. And then if you have UUU, there would be phenylalanine. So through the process of translation, you read this mRNA and you thread them with the amino acid, and this stretch of amino acid is what makes a protein. So as you can realize, if there is one change in the DNA, then it will make an altered codon, and that codon will be read as a different amino acid, and as a result of which you would have a change. So this kind of a change is what one place will lead to sickle cell or beta thalassemia harbor a huge amount of differences. So now, when people started the Human Genome Project, they thought that we would have an answer for everything, but what turned out was that we still do not know what is a normal human. We don't have a understanding of individuality, and what from the DNA sequence, how you can define health. Not only that, in the modern times, there is no definition of health unless you contextualize it by a disease. You're not diabetic, you're not asthmatic, you are not something, then you are normal. But how normal you are, that understanding is not there. And that's where we come in, that understanding of health was known to Indians thousands of years back. And this has also been acknowledged in 2016 in a very prominent journal. And it talks about, in India, the concept of Ayurvedic medicine, the goal is to maintain balance, and treatment are tailored to each person's Prakriti or constitution. And if you look at the aim of Ayurveda, it actually has an individualized context of healthy and diseased. So as a result of which, they were not talking about population, but they were talking about individuals, for understanding, the aim of Ayurveda was maintenance of health in healthy and disorder. So this is where it got interesting for me. And 2001, we started this area called, which now is being called as Genomics all across the country. But we thought that, is there a possibility to integrate such a kind of an understanding with modern genomics and see whether we do better in understanding health and individuality. Ayurveda has different ways of understanding disease. It has a way of talking about which diseases are curable, not curable, can be easily cured. It has many aspects given about physician, medicine, nurses, patient, and it also has descriptions which are similar to what we would talk about in genetics in today's time. It also has this concept of samprapti, and understanding the stages of intervention, which modern people are trying to still understand, why you have so many intermediate pathophyenotype and one is not able to understand the basis of those. And more importantly, it has this Prakriti, where every population can be binned into 7 broad constitution types. And once you are able to define a person from their constitution type, then pretty much you might be able to predict what are the likely responses in what kind of triggers, what are the kind of disease trajectory one can take, and how you can individualize management according to a person's Prakriti. And it has a systems approach, so it doesn't look at only one system, and it also is composite constitution type, which kind of tells you about Prakriti. More than that, It also has this GIS kind of a thing, so that Prakriti has the contribution of parental contribution by Shukra Shonit Sanyoge, then mother's diet, environment, lifestyle, Kalgar Bhashay, Mathurahar Vihar, and the context of Jati, Kul, Desh, Kal, Vai, and after that is the individuality. So, it doesn't have a static definition of a person. It is layered on Prakriti, and one can, once you know the Prakriti, then you know in spatio-temporal dimensions how the person is going to present, as a result of which when you see a Prakriti, you have to understand it in all these dimensions. Now, the basic thing because of which you have these Prakriti is because of these three attributes of Vat, Pitta, and Kapha, where depending on what proportions you have, you belong to either Vat, Pitta, or Kapha kind. And what is homeostatic for Vat might be disease for Pitta, and what is homeostatic for Pitta might be disease for Kapha. So it becomes very important to understand what is your baseline threshold, so that if you want to know how much you have moved from your threshold, then can you bring it back based on this concept of Trisutra, that what is the cause, what is the feature, and then you can have a appropriate therapy. So this is like a network medicine, what people are now talking about. And the treatment is brought about by a reasoning approach. So, most interestingly, all these attributes of what would qualify as Vat, what would qualify as Pitta, what would qualify as Kapha, are also given as anything which governs movement, proliferation, excretion, networking, initiation, qualifies as Vat, whatever governs metabolism, heat, thermoregulation, immune. So there are these functional attributes which are also ascribed to a Prakriti, which leads to uh, maybe and all the different cell types are also described to be abundant in whatever doshas are there, which, if it manifests more in particular organs, can maybe allow you to differentiate the features which could be attributable to Vat, Pitta, or Kapha. And so if an individual is of Vata type, maybe it would get more differentiated to certain attributes which allow you to classify people into Vata, Pitta, and Kapha. So, based on this fundamental knowledge that Ayurveda has a way to classify people, we thought that if we take healthy population and classify them by Ayurveda Prakriti, would we be able to bin them into groups? As a result of which, if you now talk about NNT, you only harm a few number of people because the other number of people might be belonging to a different category. So the question was, how does it work? So this is how I started, and this is where I we thought as to, if we want to make these two language interoperable, because there is a good science there which is needed today, then how do we approach the problem? So now, when I ask an audience, and any audience, if there is some similarity in all of them, mostly the first thing which comes is everything is A. Some people will say that it's color, but if I say that all of them are unified by certain things, it's very difficult for us to understand what it could be. But if I now tell that each of them is actually a color, so if you say absolute zero, it would mean this color, acid, it would mean this color in a particular color palette. But if I ask you to draw this color by telling you this, it would be very difficult to get to this color. And that's because you need something in the interface which will allow you to actually convert a term into a color. And then if you know this color, then you can make any other shape by the same color. So what is the missing link? The missing link is that you need a RGB code. You need a interoperable way by which it 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 understands this in a particular language which can be converted to a color palette and once you know that, you can make another one. So for me, or for us, when we started, Ayurveda was this, and we wanted to reach here. And that's where we thought that if we use the language of genomics, could we be able to merge these two by a common interoperable language? And that's how we had this working hypothesis that uh, if we think that Pith people are more prone to hemorrhagic disorders or more have metabolism enriched in them, Kapha have more storage or more obesity prone, Vata have more transport and signaling or more neurological disorder prone, then if we take people at the end of the spectra of health and look at their genome, would they differ with respect to some of these biological processes in the peripheral blood? Could we see them by RNA measurement, what I showed you? Or could they exhibit differences in their DNA level, which makes them more prone to certain kinds of disorder? So this is a very ancient primitive slide. 2001, I created this slide, and this is how we started. So we thought if we take people, so at that time I was involved in an Indian genome variation project where we were making a map, genetic map of India, and we had understood that people of particular places would be more genetically related. So we kept the jati aspect constant, we kept the age constant, and then we took people who are of one genetic background, just not to have a confounding effect that what we are seeing is not because of differences in Prakriti, but differences in the different genetic background. And then Bhavna had designed a questionnaire from where we could identify predominant Prakriti types. So if we classify them and then identify who are the predominant Prakriti and then take their peripheral blood, do we see differences? So this is what is we did. We took people, we took identified their Prakriti and then took their biochemical profiles and their genome wide expression, looked at their genetic variation, and then tried to synthesize the observation as to do we see any differences? Do those differences mean anything? And then could we correlate it back to Prakriti differences which are described in Ayurveda? So this was the first work which was published in 2008. And here we found some very interesting things. The first thing which hit our eye is that if you do a biochemistry, you have a broad range. And then we found that if you take Vat, Pitta, Kapha, they have range within a range for a particular parameter. For example, hematocrit was in the higher range for Pitta, but lower range for Kapha and Vat. If you look at lipid profile, they had the higher range. These were all in healthy. So that means if you are in this range, moving to this itself is a disease, but all of us wait for everybody to go out of range till we say that the person is diseased. So Vat, Pitta, and Kapha are a range within a range. This was for biochemistry parameter. And then what we did was we did something which is called as genomic profiling or transcriptome profiling, where we looked at, took the peripheral blood, isolated the RNA, the entire RNA, and looked at whether these RNA differ amongst the Prakriti type. And there are methods to do it, which is called as transcriptome profiling. I'll not get into the details of the method, but suffice it to say that we got different kinds of differentiating things amongst the Prakriti. For example, over expression of cell cycle stood out in Vat, immune response stood out in Pitta, LDL, HDL, lipid profiles, urea, uric acid, all those stood out in Kapha. So this was the first time we did some molecular analysis, classified people by Prakriti, and we started to see differences in people. And so this was how Ayurgenomics started. And then if you go to the Ayurveda description, then cell cycle governed by Vat is actually very, very deeply described in Ayurveda, where one talks about Parmanu Bheda, Parisankhya, Ati Bahut, Ati Sukshma. All those things are very vividly described. So this is where we say that we got an interoperable language, that if we say cell proliferation, there is a corresponding description in Ayurveda. So the knowledge starts to build up in an iterative manner. Then the question is, we found the difference. Does it make any more sense? So this came out of peripheral blood. So if you look at the descriptions of cell proliferation, that supposing somebody there are databases where you can query what happens when you have abnormality of cell cycle. So there are certain kinds of diseases like Fanconi anemia, where cell cycle goes wrong. And then if you want to go and look at the description of Fanconi anemia, you will find that it is caused by DNA damage and proliferation. And if you look at the phenotype of Fanconi anemia, then you will find that lots of phenotypes of Fanconi anemia are like Vata kind of phenotype. Now, after many years, what we asked is that this was the gross phenotype we looked at the people. How about if we take the cell derived from Vata, Pitta, and Kapha? Would they also show this cell proliferation phenotype? So now what we can do is we can take peripheral blood, we can immortalize them and create cell lines of people, which are called as lymphoblastoid cell lines. So in a different cohort, we took 3 Vata Prakriti, 3 Pitta Prakriti, and 3 Kapha Prakriti, and tried to do an experiment in the test tube in the lab. And what we found over there was, again, the proliferation rate of Vata and Pitta was much more higher compared to Kapha. So this is the doubling time. So Kapha takes a longer time to double, whereas Vata and Pitta takes faster, doubles faster. So now, for me, it was a little counterintuitive because Kapha has a bigger body size, Vata has a smaller body size. If you think about a bigger body and a smaller body, they would differ in cell number. So why would cell proliferation be higher in a Prakriti type, which is actually small bodied compared to Kapha? So what we did was we started to ask a question by meaning ask this question by doing an experiment where we gave UV to all these growing cells. And what we found was in the Vat, there was more killing. Vat cells were more susceptible to UV damage, whereas Kapha, there was no killing. But if we waited for some time after giving the UV, the number of Vat and number of Kapha became the same. So it seems to be that Vat people have more prone to DNA damage, and as a result of which enhanced cell proliferation might be a protective phenotype in Vata. So this is something which we published. This was 10 years later after we observed the cell proliferation phenotype in peripheral blood. And then I got a little curious and we found that autistic children who have a bigger head size actually have the same kind of a cellular phenotype as Vata phenotype. And this was done by actually growing iPSCs of children who have a bigger head size, autistic children, and they found that it actually is more DNA damage prone and it actually replicates more, because of which it has a bigger head size, which is called as a typically as a macrocephaly. So now in autism, this is by the modern people, they are talking about that maybe cell proliferation event in the during the prenatal stages is what gives rise to this typical clinical phenotype. And if you go by Ayurveda definition, autism is something which would be classified as a as something to manage for Vata perturbation. So this is something where now we see that from Ayurveda descriptions, from the phenotype, we actually get a cellular phenotype which has a meaning, and one can start to connect the dots between the modern medicine and Ayurveda. The other thing which we asked was that supposing we see expression differences, do they have differences at the genetic level because of which people differ in expression? And we tested some genes which differed at the expression, around 50 genes. And I'm explaining one gene which came out super interesting in our study. And this is something which is called as an oxygen sensor gene. So as all of you know, hypoxia is a condition where many diseases get precipitated, and typically you need to calibrate yourself in low oxygen condition. You go to high altitude where the oxygen levels are low, then the body moves into another phase where coping mechanisms of low oxygen switches on, and as a result of which angiogenesis happens, more blood cells are formed, you move to glycolytic mode so that you use your energy less. All those kinds of conditions come up. But now what we found was when we've taken Vata, Pitta, and Kapha and we took them at the baseline level, we find that they differ with respect to this one gene, which is an oxygen sensor gene, such that it predicted that pith people are more pro-hypoxic in condition, and as a result of which it could be predicted that they might do better in low oxygen condition, whereas cough were of an opposite kind. So, since we were doing the Indian Genome Variation Project and we had samples from Leh Ladakh, it gave us an opportunity to test this because there was a genetic variation which was connected to this difference in oxygen sensing ability. And when we did this, we found that populations which are residing in very high altitude have something which was similar to what you have in Pith Prakriti people. So then we asked a question that if this was so, then if Kaph kind of people move to high altitude, would they be the people who will have more high altitude pulmonary edema? And we had a way to test that out because we had a cohort of people who had gone to high altitude and suffered from this condition. So when we did the genetic testing, we find that the people who develop HAPE at high altitude are more like Kaph kind, and the people who are more adapted to high altitude are more like Pith kind. So this was the first time we could actually show that if you screen people without exposing them to low oxygen condition, you might be able to better predict who are the people who are likely to do better at high altitude, and as a result of which this could be of enormous use to people who take sojourns to high altitude or army people, like they are deputed to high altitude conditions. Now the question is, does this kind of observation of pith acclimatizing better, cough having a problem at high altitude, does that match the descriptions of ancient texts? And there we found that in Sushruta, you have diseases described where cough vat is more prevalent in high altitude. So having a pith kind of a prakriti might be much, much more suitable in high altitude conditions. So this was one of the ways by which we could show. And what was very interesting is that science has to be reproducible. And this kind of a observation of the gene in high altitude has now been shown in multiple altitudes of the world. And very recently in Quechia, which is again at high altitude, people have shown that the same gene is linked to high aerobic capacity. So this shows that the power of the way to phenotype people can allow us to discover predictive marker just because there are susceptibilities linked to these different phenotypes. And this gene has become a very important therapeutic target because wherever there is hypoxia, either you need to stabilize, meaning when you have ischemia, you have hypoxic condition. When you have cancer, you have hypoxic condition. So EGLN1 has become a very important therapeutic target. And so you can imagine that if inherently there is differences in these people with respect to EGLN1, then all these kinds of multi-system phenotypes might differ among the individual, so that some people might have better recovery after ischemia, some people might repair cartilage better, and some people might have more susceptibility. Now, based on this, colleague Anurag, he works in asthma. He decided to test this out in asthma. So the question was, if you have a mouse model of asthma and you modulate it by modulating EGLN1 so that you make it more pro-hypoxic, what happens to the mouse? So he tested it out, and he found that if in asthmatic condition of mouse, more hypoxia sets in, then it causes inflammation and epithelial injury. And it is so bad that there is a steroid resistance which sets in, mitochondrial shape gets perturbed, mitochondrial function gets perturbed, and which shows that inflammation can exacerbate asthma. And maybe there's a possibility that if you are Pith Prakriti having asthma, you might have more inflammatory consequences which have to be managed differently. But what it gave us was something very interesting, that if asthma condition can be recapitulated in mouse, where you give a pith cough, pith elevating condition in asthmatic condition. Now you take a medicine from Ayurveda, which is used to modulate pith cough in asthma. Would it work in this mouse model, and could it reverse asthma? So this was what was tried by this medicine Adatoda Vasika, and Bhavna led this work. And so we gave Adatoda Vasika to the same model, and we could show that it not only reverses asthma, it makes it more steroid sensitive, and also all the mitochondrial shape, function, all what had got compromised is alleviated. So this actually, by this kind of a logic of Trisutra, one can understand the molecular mechanism of Ayurveda drug. Not only that, when the COVID came and there was a hypoxia which was very, very relevant, so we tested this out in preclinical model, Adatoda Vasika, and we found that all the preclinical conditions, COVID-19 like condition, Adatoda Vasika was useful. So as a result of which, now you can see if you have a Vyadhi, which is Anukt Vyadhi according to Ayurveda, how foundational principles of Ayurveda can allow you to evolve a medicine in a rational way, and having a connect with the modern signature allows this to be generated even in a better manner. So this actually shows that this could be not only state of the art, it could be beyond the state of the art, because this was also tried out in a COVID trial with All India Institute of Ayurveda. Now, now the relevance of Ayurveda became even more apparent during the COVID time, because what was the WHO definition seemed to be something which has been actually talked about of in Jan Padudvans so many years back. And if I've done this in thousands of people now, and the same kind of data pans out that many people availed Ayurveda medicine either by way of prevention, by way of treatment, or by both ways during the period of COVID. So as you can see, hypoxia response hemostatic outcome, which we discovered through this Ayurgenomics approach, became more relevant during COVID time, where you see oxygen compromise condition, hemostatic condition were very evident. So it would have been worthwhile to test whether different Prakriti people have different kinds of exacerbation when you have such kind of an infection. Now the question is it beyond the state of the art? So we also developed another framework. So for example, this Hetu Ling Aushad worked in the earlier case, that we started with Ling, we know the cause as hypoxia Hetu, and we could evolve a medicine. So if this is internally ratified, then if I start with Aushad, can I actually find molecular basis and the causes? Can I connect them using the genomics approach? So this is the next thing we tried, because then if we are able to do this, develop this platform, then we might be able to repurpose Ayurveda medicine even in modern medicine condition where the features and causes are described in genomics language. So what did we do? We took a plant, Schizamplus pereira. It's used for treating snake bite, hormonal disorder, febrifuge, sepsis. So very unrelated condition for a modern person. But Ayurveda doctor uses, even African folklore is described that these are used in very, very contrasting condition. A very interesting trivia is that wherever malaria is endemic, Schizamplus pereira is also very prevalent over there, which also kind of resonates with Ayurveda descriptions about the presence of medicinal plants around the place where the disease most abound. So now the question was that there are so many things where hormones play a role, and then there is a viral inhibition. So is it that Schizamplus pereira has so many molecules that some things are useful for viruses, some things are useful for estrogen, or whether there is a common unifying link because of which it is used for very contrasting conditions and can be used by the same doctor. So what we did was we used a framework. It's called connectivity map. So MIT Broad has actually developed a platform where many drugs are millions of drugs or millions of small compounds are treated through cell lines, and you see what genes go up and what genes go down by one perturbation, second perturbation, third perturbation, and like that you get a pattern that so many genes have gone up, so many genes have gone down, so you generate a pattern of all the drugs. So now what one can do is you take a new drug treat it with the same cell line, generate a pattern, and match it with the known patterns. Now, if these drugs have a known mechanism of action, then one can actually start to connect whether this unknown drug has a match with a known drug. And if the known drug has a known mechanism of action, then you can start to speculate what would be the likely way that this medicine works. So what we did was we used Sesam plus Pereira and took it through the same pathway. And we could actually find that it works like a protein synthesis inhibitor. Many of them are used in dengue. So we tested it out in dengue cell line and we found that it actually can inhibit dengue titer if you give Sesam plus Pereira. So this was one part. But the second part was the estrogenic link. So what we found was that even CEPA at the transcriptome level gives signatures which are hallmark of different kinds of breast cancer, which are recalcitrant to different therapy. And this is because the estrogen receptor goes down. And then uh, we looked at this estrogen receptor and we looked at CEPA, and we found that multiple molecules of CEPA bind to certain pockets, and some of these pockets are the places where tamoxifen and beta estradiol, the more prominent medicine for breast cancer, bind. So it seems to be that CEPA could be modulating estrogen, and not only that, if the estrogen is modulated, then the dengue inhibition also is not so well done. So that means there is a link between the estrogen axis and the viral inhibition axis through the cholesterol or lipid synthesis pathway. During the time COVID struck, and we thought that could this be repurposed to COVID condition, because we saw some patient samples, we found that whatever transcription signatures were up were down by this drug. And then what we did was we tested out the CEPA in cell lines, and we took also single molecules from CEPA. And what you can see is that if you take full extract, the root extract or the hydroalcoholic extract, the titer is down by 99%, which is not done by all the single molecules. This is the control. So this showed that during the time of CEPA, if this connectivity map signature is done during the time of COVID, one could repurpose it for even COVID-like condition. But it was not done during COVID. Very hardly any people, any doctor would have used CEPA. But after that, when we got this result, I asked a couple of Ayurveda doctors. They said that if such a kind of a knowledge is also available, one can actually test this also in COVID-like condition, and it would match to descriptions which are given in Ayurveda. Not only that, I was very pleasantly surprised when one Ayurveda doctor who had heard my lecture said that in Ayurveda there's something called Sthanya Shodhan, which is linked to Sesam plus Pereira. And maybe there's a possibility that it could also mean that it might be useful in breast cancer, but it is testable. So this is where this molecular yukti comes in, that it's an iterative language. It's just an interoperable language of Ayurveda aspect converted into a molecular language in such a manner that maybe an Ayurveda doctor can also use this to build more evidence or more understanding of whatever is given in Ayurveda. So to summarize, how we first developed a working hypothesis based on Tridosha Trisutra principle, tested it out in Prakriti and cell line, and got multi-omic correlate and links to doshas. And once we got it, we found whether there is a genetic basis to these links. And we found that there is a genetic basis, and that is what explains differences in phenotype, differences in susceptibility, differences in response. So now, once you find this genetic link, you can perturb that in a mouse model so that you can study in much more detail. And that's where which becomes a model system for dosha modulation and Vikriti. Once you have that model, which recapitulates what Ayurveda talks about, then you can use Ayurveda herbs from their dosha modulation action and try to test out whether if tridosha is a common organizing principle, then this could be, you could understand the mechanism and then you can use it for repurposing. So this is the entire molecular framework of Trisutra, which starts from the foundational principles of dosha. So whatever we have done, many people have now got evidence of at different level, at the cellular, genome, epigenome, transcriptome, proteome, metagenome, biochemistry, that at any scale or any hierarchical scale of cellular or physiology, if you classify people by Prakriti, there are differences, and these differences can allow you to stratify people. And then this could become a very good way to have a non-invasive diagnosis or develop better integration with modern condition. So, so as you can now understand, genomics can be understood to understand phenotypic individuality. It can give us an idea about genetic individuality. It can give us molecular correlates of dosha. It can be used to understand molecular mechanistic basis of dosha. It could give us new biomarkers and actionable target. It could help stratify people for looking at their health outcome, and then repurposing possibility, or also allow us to understand the mechanism of how wide if people would differ with respect to drug action. But more importantly, we can now start to think about how two doctors, modern medicine and Ayurveda people, can now cross talk with each other, and we have a more integrative approach to medicine. So this is what we are doing now in Ayurtech here, where we are having these foundational principles, how we can connect it with different kinds of engineering sciences to be able to translate these concepts into a clinical setting. So what we are doing is we are taking the principles and practices, integrating it with data science, engineering, and multi-omics to be able to see how we can port this through the common unifying language into interfaces which could be linked to ABDM kind of platforms, and we could have interfaces. So this is a team of interdisciplinary people who are working with along with Ayurveda doctor. AIMS also is our validation partner, and we are trying to see how we can use this to leverage the system's understanding, preemptive management, understanding disease trajectory, understand unique therapeutic modalities, and then try to understand the context of population, culture, and habitat on individual baselines. So we are 2 years in the project in Ayurtech over here. So we are using different kinds of methods. I'll not go into the detail. That's a lecture of another time. But we are using now many different AI-based approaches or phenotyping people on the basis of Prakriti. We are doing big data analytics. We are trying to classify or get ontological descriptors of through the big data, and then many engineering things are kind of attempted. And we are also training a large number of Ayurveda graduates over here during with workshops and all, where they can get oriented to all these aspects. So finally, I would like to thank all of these people, and from where we started at Trisutra at IGIB. And these are all the collaborators over the years I've had. So now I'm open for uh, questions. I hope I have not gone too specialist in genomics, and I hope I've been able to kind of excite you people to ask a few questions. Thank you.
Thank you so much, madam. It was really a very excellent lecture we listened today, and we could see the concept of Prakriti in very different perspective. We never thought some of the areas like you mentioned about the different altitude and the response of the Prakriti in different manner. So it was really a very excellent explanation, madam. So participants, they are encouraged to put their queries in the chat box.
Mm-hmm.
So there are a few questions, madam.
Okay, let me see. I'm not able to see the chat box. If you can read out the questions, I can answer them. Uh, I'm not able to see the chat box.
Uh, there is one question from Ranga Shri. How can Ayurveda doctor join such institution and work for this field?
Okay. So what we are trying to do with Dr. Saket itself, we are trying to build a virtual platform for crowdsourcing information. We are trying to call it as Anukta Collective. And the idea is that there's so much information in the modern literature, and if you are able to be geared up to read that literature and connect to Ayurveda sciences, you can start to develop conceptual concordance. And we did NCISM supported some postgraduate students, around 90 students. They came in 3 batches of 5 days each, and we gave them intensive training on technologies, Ayurtech, along with, and then they had Ayurveda doctors, mentors. So the whole day they would have lectures, and in the evening they would be oriented to Ayurveda concepts, which connects to these technologies. And in the fifth day, they had to make a presentation as to how do they think that the technology connects to their sciences by way of understanding mechanism, by way of having modernization of certain therapies or whatever it is. So so we formed a cohort, and we encouraged them that if you have an MD problem which requires a technology, and you can get in touch with us and we can help you out. There are lots of faculties in different engineering department. So we have a center of excellence which is supported by Ayush, and we hold workshops where people can come, and you can work in projects if we have funding. At this point of time, our funding is getting over, but we are also creating some kind of a virtual space where students can engage with us and learn it during the course of whatever they do. And then one can always write projects if you are faculty somewhere, and we can mentor you through it.
Thank you, madam. There is one question. Like if we have people with Prakriti, like Dvandaj Prakriti, there is one
Yes,
single domain, Dvandaj
yes.
Prakriti and Sama Prakriti
Uh-huh.
also.
Yes,
So
yes.
how we can understand this?
So this is the most challenging thing. So at this point of time, we used predominant Prakriti as a window to doshas, right? And now we want to understand whether when you have Vat Pith or Vat Kaph, whether they actually are some, or they actually balance out each other, because of which very only 10% of the people are of predominant Prakriti, majority are of the Andaj Prakriti. So now, like, I think, and this is my working hypothesis, that if you have a pith cough personality, normally a pith or a cough will be more vulnerable to diseases. So if you have elevated pith, cough is your more harmonizing medicine or nutrition or whatever it is. So now at the prenatal stage, does the cell understands that? And so supposing you have proneness of pith from certain set of gunas, do the cuff people have an opposite set of gunas? So if you have pith cuff, then is one is balancing out the other, then which is the susceptible phenotype? So for this, we need to study large number of people. And if you want to study large number of people, you need to classify Prakriti in a very objective manner. And that's where now we are using this computer vision so that we can scale up Prakriti evaluation in thousands of people. So what we are doing is Prakriti has Prashna, Sparsh, and observation, right? So we are converting the observation by cameras, by many devices, AIoT, so that whatever Ayurveda doctor sees in a person, darshan part, we are converting it into visual devices, and then asking for other kinds of questions. And if we are able to reduce the number of amount of time and increase objectivity, then we would have huge data set to answer the questions which you have asked. That what happens to Dvandaj Prakriti, how are they prone, or how are they habituated, or etc. So this is not studied as far.
Yes, madam.
Okay.
Yeah,
Uh,
chat box.
what will be the sites of research on these contexts? I did not understand what is
Uh,
the
yes, madam.
question. Sites of research means? I did not follow
Question.
this.
Yeah,
Somebody,
question is not clear, madam. And then another question is: are there any possibility of Prakriti-based drug designing for a specific disease using aerogenomics and aeroinformatics network pharmacology?
yes. So my colleague over here, his name is Dr. Sudipto Bhattacharya. He's actually doing it. In fact, he has crystallized a protein out of Triphala, which has very interesting wound healing properties. So he's looking at the polypharmacological perspective. We are developing platforms over here for polypharmacological properties. And so, for example, prameha. Prameha, you have pith modulated prameha, kaph modulated prameha, or vat, meaning vat prameha, pith prameha, kaph prameha. They might present differently in different prakriti types, and they might require different drugs. So we need to understand people, the prakriti of people who have prameha of different kinds, and molecular mechanism of action of different drugs which are used for different doshas. And then we have to do a proper clinical study of prakriti versus dosha. So these are all the questions which we are now trying to do. So this requires tools, and at this point of time, such kind of tools are not available. And I do not think that the modern approach or modern tools can be just ported to address an Ayurveda question. There are lots of methods, but they have to be used from the principles of usage of Prakriti or principles of Prakriti, not Prakriti, Ayurveda, dosha concepts or whatever concepts. So those are the frameworks which we have to develop and we are developing.
Yes, madam. Dr. Darshini, she wished to know which particular scale was used for Prakriti assessment.
So for the predominant Prakriti, a questionnaire was developed by Bhavna, and then we had an objective scoring based on which we could distinguish the predominant Prakriti, and then we use some AI and all to be able to show that even if we remove the Ayurveda labels and just take the questionnaire and do unsupervised way, we are able to recapitulate the connections between the phenotypes within a Prakriti. All that was done on predominant Prakriti. The models have not been built in with mixed Prakriti types, and that's what we are trying to do here.
So, like, madam, one question is there by Dr. Pallavi. She wished to
Mm-hmm.
know, like, we have this Sharir Prakriti, we have the Manas Prakriti. So, Manas Prakriti
Mm.
and aerogenomics relation, whether.
Has not been attempted.
Okay.
Sharir itself is not clear. So CCRS has one questionnaire, IGIB has one questionnaire. So now with CCRS, we are trying to develop and automate this questionnaire. And then we would like to do it in large scale across the country. And that's one project, one very ambitious project we are now attempting, or we have put in a proposal. So this needs a very interdisciplinary set of people, cohorts, and it has to be tried out in different desh, kal, way, to be able to have a very, very sound model of Prakriti. Ayurveda doctors do it in a very well manner, but how do you make it objective so that many people can follow the same harmonized set is what we are trying to develop.
Dr. Kavita Agrawal, she wished to know how AI, artificial intelligence, can help in this
Okay.
area. Yeah.
So AI is we are using AI in various ways. One of the thing is computer vision. What AI does is that supposing you see a person and you use a questionnaire where you look at anatomical attributes of the person and you say tall, medium, disproportionate, proportionate, eye is large, forehead is large, all those kinds of things. Now what you can do is you can take a picture of the person. Once you have the picture of the person and you have Ayurveda doctors putting a label by their own assessment, then you can get the features extracted out of the picture and train models so that it's able to understand when Ayurveda doctor says this is what the picture, from the picture you can understand that if Vat is there, then these are the features which describe Vat. So that's what is called as computer vision. So that's an AI based way. Just like if you see you have pictures in Google Drive or Google Photos are there. If you have a childhood image and a image of a person at a later age, it will show both the images together and ask that is this person. So how do they make out two same person from two different age groups? So this is where computer vision comes in. So that's what we are using, and it will reduce the time to measurement, and whatever subjectivity is there in the person can be done away with. So there we are using voice also. So we are asking people to speak and getting their audio recording. From the audio recording, we are using deep learning methods to be able to understand Prakriti. So these are all AI in phenotypic capture. Then we are using AI and natural language processing. So we are using natural language processing to bridge the ontology if Ayurveda, Sanskrit language with English. So now you see Google has or chat gpt, you write something in English and ask it to give it in Hindi, it'll give it. So it has to understand the semantic, it has to understand the usage and connect it. So we are using Ayurveda knowledge and connect threading the two languages together. That's where AI is also getting used. We are using AI for understanding so many drugs are there and so many molecules are there, meaning targets are there. How would you find them out? So there are many different places where AI is getting used, and that's why our center is focusing on how to use AI to leverage all this to scale it up. Basically, AI is used once you have a knowledge, then you can scale up a whole lot of things, but you have to build the knowledge, and that knowledge we first have to build from Ayurveda perspective.
Yes, madam. There is one question about the what are the biomarkers for
Mhm.
application in Ayurgenomics?
So we have done till now only foundational studies, and biomarker will come out when you are studying a disease, right? So that those are the studies which are now going on, and hopefully in a couple of years we might be able to get. So EGLN1 is one biomarker, very interesting biomarker. It is used in multiple scenarios in hypoxia. There are other markers we have done, exome study. We have done other kinds of studies where we have also got many other genetic variations which differ between Prakriti types, and we need to study them in clinical settings to be able to say whether they could be biomarkers of certain things. So for that, we have to implement these in Ayurveda settings, and Ayurveda doctors have to come forth to test them out.
Yes, madam. Dr. Astha Parmar, she is asking the learning resources, some
Uh-huh.
books or other things.
So there are lots of papers now. At this point of time, there is no book on this, but there are lots of publications, and I can share the whole folder, and then you can share it with, meaning I can share it to the mail ID from where I got the invitation, and that can be forwarded to all the
Yes,
participants.
madam. Please, madam. Even you can share through the WhatsApp. So we have
Sure,
created Google Classroom for this particular translational Ayurveda. So we can
sure,
share those Google Drive link to the participants.
sure, sure.
So they can easily avail all the resources. Dr. Preeti Parab, she wished to know, is there IGIB Prakriti analysis tool training? Whether training is available for
So
Ayurveda
I
students?
am not in IGIB anymore. I'm in IIT. So I'm totally meaning I have taken retirement from there. So I'm now IGIB. Dr. Bhavna is taking care of it. So I do not know the status of that tool, but we are developing this AI based kiosk. So idea is that if we can build assistive technologies where a person comes in, you take the picture, you see the gate and collect the gate information by sensors and hear the voices. And after that, Ayurveda doctors ask the prashna component and sparsh component is assessed. And then based on AI, one can predict the prakriti of a person. So that's what we are attempting here. And we are now working with CCRAs and planning to validate it in large cohorts.
Yes, madam. There are a few very different questions. Like one question is there, the job opportunities and other things in this particular field. If someone is working, so that is one question.
So, uh, Ayush has to give that job opportunity. At this point of time, you can have entrepreneurship options, and whatever Ayurveda practice you are doing, you can actually start to integrate technologies there, right? There are lots of technologies. Unfortunately, uh, most of those technologies are catering to the way a modern doctor sees, but those can also be pivoted to Ayurveda problems. So it has to be, I think, more of entrepreneurship places. And if Ayurveda institutes like All India Institute of Ayurveda and all set up processes where they can integrate technologies, definitely there would be more better job opportunities, because there is a thrust of AI in Ayurveda and technology in Ayurveda, and that's why Ayush is supporting lots of IITs. So one has to figure out, and that's what I'm talking to Ministry of Ayush, that we should have more capacity building, more workshop opportunities, more courses on portal, so that today's Ayurveda doctor can get geared up with modern technologies and they can start to think of how to integrate the technology. So this is something which is beyond my
Yes.
scope at this time.
So one question is there AI app which include information about different biomarkers of Ayurveda and drug selection protocol through
No,
Ayurveda
no, it has to be developed. It has to be developed. See, this area is we just develop this area. It's a just an interoperable language. It's like how you do Hindi subtitles for an English movie or English subtitle for a Hindi movie. This is genomic subtitle for an Ayurveda literature, right? Now you have to make your own films and use this. So it's just that the foundational knowledge or the foundational logic took 20 years to iteratively build this, and there are not many people who have traded in this area. So we now need more number of people to participate, and then only this area will grow.
Thank you so much, madam. These were the few questions. Uh,
Okay.
Still, there are a few questions like particular disease and its genomics. So,
Mm-hmm. What is that question?
is Jogan syndrome genomic?
Joubert syndrome.
Yeah.
Huh.
So,
So
how?
we have recently done a very big exercise where what we have done is there are 12,000 rare diseases. This I have done in IIT Jodhpur. I did not explain that because it would have become too much for you people. But that paper is also there in bioarchive and it's undergoing revision. So I understood a very interesting concept in Ayurveda which I did not know for 20 years, something called Anukta, which you all know very well, but I think it is not operationalized as is. But how would it be that if you take a rare disease and try to understand, and an Ayurveda doctor, how will they look at it? And could they use the Anukta framework along with genomics? So, what we did was there are 12,000 rare diseases, and these 12,000 rare diseases, it is cataloged in a particular database called Human Phenotype Ontology. And for all these rare diseases, phenotypes are mentioned, meaning each rare disease will have 17 to 20 phenotypes, which are like short stature, this, that, and all. So we had a team of Ayurveda doctors. They labeled all these features into Samanyaj Vikar and Nanatmaj Vikar. And then what we did was we started to re-annotate all these rare diseases and said that if it were be to be looked from Nanatmaj and Samanyaj Vikar, how will Ayurveda doctor see these rare diseases? And then we used a mathematics person. She did the clustering, and we found that rare diseases which are fragmented by organ-specific manner, we could cluster them based on Vat Nanatmaj, Pit Nanatmaj, Kaf Nanatmaj, Vat Pit Nanatmaj, and Samanyas of the same kind into 6 broad groups. So now, if you say that 1,000 rare diseases have a Pitta Nanatmaj component, that is, there is a more inflammatory kind of uh, axis which governs many of these diseases, then could Ayurveda doctor look at these diseases and see whether if even they are Adhya and Yapya or whatever manageable, could you manage the pith perturbation in them so that the quality of life is maintained? So, like that, we found that there was a cluster of cough, which was all linked to celiopathies. And celopathy is something which takes 8 years to diagnose in modern settings. But Ayurveda, when we look at it from Ayurveda perspective, it all comes and clusters as a cough celiopathy. Some of them are Vat celiopathy. If it is Vat celiopathy, these are skeletal and muscular celiopathy. If it is cough celiopathy, these are endocrine celiopathy. So what I am now proposing is that if a rare disease comes to an Ayurveda clinic, could they be diagnosed through the Anukta framework, and could there be better way to manage a rare disease in a patient? So such kind of newer things are coming up.
Yes, madam. There is one more question, madam. Like, can we develop connection between the Ayurveda concept of Gunya and genomics? Gunya is the inherent weakness or susceptibility for the disease. So, whether is
Ha
it
ha ha.
possible to develop connection?
So one can look at, see if this is inherited, then one can look at the genetic basis. So supposing this runs in the families, and you collect a few families where parents are also of this kind, whose children are also of this kind, then you can do a family-based genetic study and identify the genetic locus, which always segregates with the people who have this phenotype. This is one way. Or otherwise, you take hundreds of people who are very neatly classified as this or a control group and do a genome-wide study. So these are the approaches by which maybe you can study and see differences. And this is typically how modern people study the genetic basis of diseases and identify genes which are involved in certain conditions.
Yes, madam. Thank you so much, madam. We feel to listen more and more from you because this is a very new way we are understanding the concept of Prakriti.
Uh-huh, sure. I hope it was okay for everybody.
Yes,
So,
thank you so much, ma'am. This is Mohan Joshi here.
okay, okay.
Uh, Thank you
Thank
so
you.
much. And we were actually waiting for your session, and hopefully we could complete it within the hour time frame. Thank you for
Ah,
joining, ma'am. And definitely
okay,
it will be with the students, and all recordings will go online, and they will go in archives of IAP. So anybody can visit
okay,
it afterwards on the YouTube. We will share the links, ma'am. Thank you
okay.
so.
That would
Thank
be
you.
great. Thank you so much.
Thank you
Thank you
so
so
much,
much.
madam. Kindly share the resources Google Drive link, madam, so
Yes,
that I can share
yes.
with. And
I
there
will
are,
share.
please, madam. There are so
I
many
will.
participants. They are thanking for this excellent lecture, madam. So,
Okay. Thanks to all the best. And I hope some of them get excited to take up this and move in a different direction, which actually puts Ayurveda's cognitive framework for addressing today's health problem. I feel uh, the Anukta understanding and the Yukti way of evolving medicine is one of the things which we can leverage, and especially in changes in the climate change era, where all our physiology is shifting, how to cope with the change scenario. Ayurveda might have a lot to offer. It is awaiting all the younger people to take it up in a more challenging manner. So,
thank you.
yeah, my mail ID, you will get it in the net. It's Mitali at iitj.ac.in.
Yes, madam. I will share your email ID to the participants, madam. So if they have
Sure.
any specific query, they can directly
Yeah.
connect to you. So
Okay.
once
Thank
again,
you so
thank you
much.
so much, madam, for sharing your valuable time and your experiences with us, enlightening us with the concept of Prakriti. Thank you so much, madam. Thank you so much.
Thanks. Thanks.